So, imagine you decide to install this, like, state-of-the-art, fully AI-powered security system to catch a burglar in your house.
Okay, sounds like a good idea.
Right. It's top of the line, and it detects a break-in instantly, which is great.
Right.
But instead of just locking the doors and, calling the cops, the system decides the absolute best course of action is to flood the living room, set the kitchen on fire, and completely dynamite the roof.
Wow. Yeah, that's a bit of an overreaction.
Exactly. I mean, the burglar is definitely gone, but unfortunately, so is your entire house.
Right. It's this profound failure of proportionality. The defense mechanism just initiates a total scorched earth policy.
Yeah.
It's basically operating under this fatal assumption that neutralizing the threat is worth the complete destruction of the host environment.
Which brings us to today. Welcome to Deep Dive Lab, where we break down and dissect cutting-edge scientific research, from advanced biomedical technologies to breakthroughs in AI, material science, chemistry, and physics. And we tell it all as compelling, easy-to-follow stories.
I love that.
Yeah, and today we're taking a deep dive into the human body, and we're asking a really strange, honestly very dark question.
Which is,
Why does the human body sometimes do exactly this to itself?
yeah, it's a terrifying question.
Right. We're looking at this biological mystery that has baffled and terrified doctors for decades, where patients don't actually die from a virus or a bacteria, but from their own internal defenses just going completely rogue.
Yeah, and to really understand the mechanics of this, we have to look at the baseline functionality of our immune system. Specifically, we need to talk about these proteins called cytokines.
Okay, cytokines. I know a lot of people who follow biology know that cytokines are basically the immune system's chemical text messages, right?
Exactly. But it's crucial to understand their normal operating parameters. Like when innate immune cells detect a pathogen, they secrete these proteins to call in heavy-duty cellular backup. Things like macrophages.
The big guns.
Right, the big guns. And under normal circumstances, this inflammatory response is highly localized, and it's rapidly shut down once the threat is neutralized.
So the body handles the breach and just returns to homeostasis, normal stuff.
Exactly.
But history shows us this system has a terrifying failure mode. I was looking through the historical context in our sources, and the first time doctors really gave this a name was back in 1993.
Yeah, that was a major turning point.
Right. They were observing bone marrow transplant patients, and they saw this baffling, incredibly life-threatening hyperinflammatory reaction.
Yeah.
The patient's immune systems were essentially locked in hyperdrive, indiscriminately attacking their own organs, and they called it a cytokine storm.
Yeah, those 1993 observations were a watershed moment because the clinicians were watching this systemic cascade that they just couldn't stop.
Which is terrifying.
It really is.
What?
And since then, we've identified this exact same storm pattern across a massive spectrum of seemingly unrelated conditions.
Right, like where else do we see it?
Well, it happens in severe autoimmune diseases like lupus, genetic disorders like hemophagocytic lymphohistiocytosis.
That's a mouse.
It is, yeah, HLH for short. And we see it in severe sepsis, and it became notoriously recognizable during the COVID-19 pandemic.
Wait, hold on. Okay, let's unpack this for a second, because I'm looking at the biological half-life of these cytokines in the source material.
Right.
These proteins are biologically designed to degrade in a matter of minutes, right?
Yes, very fast.
They have an incredibly short half-life, precisely so that inflammation resolves quickly. So, I mean, if the original virus or bacteria is already dead and the cytokines naturally fizzle out on their own, what is keeping the storm going?
That is the million-dollar question.
Right. Why doesn't the alarm just naturally stop ringing once the primary threat is gone?
That is the exact paradox that stalled immunologists for decades. I mean, if the messenger molecules degrade in minutes, the cellular response should naturally quiet down.
Exactly. It doesn't make any sense.
It makes no thermodynamic or biological sense for a patient to progress from a standard fever to lethal multi-organ failure if the initial trigger has already been cleared out.
So where is the signal coming from?
Well, the continuous, overwhelming supply of cytokines had to be coming from somewhere else, but no one knew what was driving the production.
Until quite recently. Because it wasn't until 2026 that a team of researchers finally found the missing puzzle piece.
Yeah, a massive breakthrough.
Which brings us to this massive unifying study published in the journal Nature Review's Disease Primers. And the research was spearheaded by Dr. Rajendra Karki, Dr. Thrumala Devi Kanaganti, and their colleagues at St. Jude Children's Research Hospital, along with other global institutions.
Right.
And what's wild is they didn't just look at one disease. They mapped out the entire underlying matrix of this phenomenon.
Yeah, and their findings directly address that half-life paradox you just brought up.
Okay, so what's the answer?
Well, it turns out the short-lived cytokines are not simply lingering in the bloodstream. They're actively triggering a lethal, self-amplifying positive feedback loop.
A feedback loop.
Right. When the local concentration of cytokines reaches a certain critical threshold, the immune system actually stops perceiving them as mere text messages.
Wait, what do you mean?
It begins interpreting the overwhelming volume of cytokines as evidence of catastrophic tissue damage.
Oh, wow. So the cytokines themselves become the danger signals.
Exactly.
I was trying to picture this, and it feels like an exploding alarm clock.
Oh, I like that.
Yeah, like it doesn't just ring to wake you up, it physically blows up, and the shrapnel from that explosion hits the alarm clocks next to it, causing those to blow up too.
That gets the explosive part right, absolutely.
Mm.
But to be scientifically accurate, it's less about the noise or the explosion itself and more about how that shrapnel acts as a physical key in a lock on the neighboring cells.
Okay, break that down for me.
So the 2026 study highlights what happens when two very specific cytokines, tumor necrosis factor, or TNF, and interferon gamma, act together.
TNF and interferon gamma. Got it.
Right. And their synergism forces those neighboring immune cells to undergo a highly violent inflammatory form of cell death known as PANoptosis.
PANoptosis, that is a heavy term.
It is.
Break down the mechanics of that for us, because usually when a cell dies naturally, it's a pretty quiet process, right?
Yes, exactly. Standard cell death, which is called apoptosis, is incredibly tidy.
Right.
The cell neatly packages its internal contents into little vesicles for disposal by macrophages, which are basically the garbage disposal cells of the body.
So it cleans up after itself.
Exactly, doesn't cause a scene. But PANoptosis is a lytic pathway.
Lytic, meaning what?
The term lytic means the cell membrane is physically ruptured. The cell bursts wide open, spilling its internal contents directly into the surrounding tissue.
Oh, yikes.
Yeah, and this includes newly synthesized inflammatory cytokines and what immunologists call DAMPs, damage-associated molecular patterns.
So literally the biological shrapnel from the alarm clock.
Exactly that. Now, how do the neighboring cells actually feel this shrapnel hitting them?
Right, how do they know?
Well, they possess innate immune sensors inside their cytoplasm. The researchers identified specific internal tripwires, such as a sensor called ZBP1. Yeah. And ZBP1 is fascinating because it specifically detects something called Z-nucleic acids.
Wait, Z-nucleic acids, are those different from normal DNA or RNA?
Radically different. Normal DNA forms that classic right-handed double helix we all recognize from textbooks, right?
Right, the classic spiral staircase.
Exactly. But under extreme stress, or during viral replication, genetic material can actually kink into an abnormal left-handed zigzag shape.
Whoa, really?
Yeah, that is a Z-nucleic acid. And when a cell ruptures and spills these abnormally folded molecules, the ZBC1 sensor in the neighboring cell immediately recognizes the wrong shape.
It's like it knows something catastrophic has happened next door because the DNA is literally bent out of shape.
Precisely. And its response is brutal. Upon detecting these Z-nucleic acids or other DAMPs via sensors like NLRP3, the cell begins assembling a massive multi-protein complex.
Which is called?
It's called a panoptosome.
The panoptosome. That sounds intimidating. What does it actually do?
Think of the panoptosome as a biological scaffolding, or essentially a microscopic guillotine that the cell builds to execute itself.
It builds its own guillotine.
Yes. And once the scaffolding is fully assembled, it activates executioner enzymes called caspases.
Caspase.
Okay. And these caspases act like molecular scissors. They physically slice through the structural proteins that hold the cell's membrane together.
Oh, wow.
Yeah. They punch actual physical holes in the cell wall. So the neighbor cell rapidly fills with fluid, bursts, and then releases its own payload of DAMPs and cytokines.
And the loop is locked in.
Mm-hmm.
Cell A ruptures, spilling Z-nucleic acids. Cell B detects them, builds a panoptosome, uses caspases to punch holes in its own membrane, and explodes, triggering cell C.
Exactly.
It's a massive chain reaction of cellular lysis, completely independent of whatever virus started the whole thing in the first place.
Which is exactly why the storm doesn't fizzle out. The immune system is feeding itself with its own destruction.
Okay, let's pull back from the cellular level for a second and translate this into what is actually happening to a person.
Good idea.
Because if you imagine you're standing at the bedside of a patient experiencing this, the research maps out a highly specific, terrifying, real-time pipeline of this storm progressing through the body.
Yeah, and the speed of the deterioration is what really catches clinicians off guard.
Right.
So days 1 to 2 represent the onset phase. If you are looking at the patient, they might just have a fever, some chills, maybe some muscle aches, and mild tachycardia.
So it perfectly mimics a standard, robust immune response to an aggressive infection. Nothing too crazy yet.
Exactly. But beneath the surface, that PANoptosis loop is just starting to warm up.
And then we hit days 2 through 5.
Right. The amplification phase. This is when the chain reaction really accelerates. The research notes this is when patients start experiencing severe hypotension, which is a massive, dangerous drop in blood pressure and vascular leakage.
A vascular leakage, that sounds like a really critical turning point.
It is. Think about the endothelial cells. These are the specialized cells lining the inside of your blood vessels, kind of like a biological Teflon coating. When the cytokine storm hits them, those tight junctions between the cells break apart. The blood vessels literally become porous.
Oh.
Yeah, fluid begins leaking out of the bloodstream and pooling into the surrounding tissues.
And that leads directly into days 5 to 10, right? The peak or crisis phase.
Exactly.
This is where the vascular leakage causes mechanical failures across the entire body. And the most infamous one is acute respiratory distress syndrome, or ARDS.
Yeah, ARDS is basically a mechanical suffocation. Because the blood vessels in the lungs are leaking, fluid floods the alveoli, which are the tiny air sacs where oxygen exchange actually happens.
So they just fill with water.
Right. The sacs fill with water, meaning oxygen physically cannot cross into the blood.
That is horrific.
It gets worse. At the same time, the systemic inflammation triggers neurotoxicity, leading to confusion or even seizures. The blood coagulation cascade goes haywire, forming microclots everywhere.
Wow.
And the leaking vessels mean the heart can't maintain blood pressure, so vital organs are basically starved of oxygen.
So going back to our analogy, the AI security system hasn't just flooded the living room. The roof has completely caved in and the foundation is cracking.
Perfect analogy.
Right.
And if the patient survives that physiological earthquake, they face one final hurdle.
Which is?
They either enter a slow resolution phase or an immune exhaustion phase. The immune cells have destroyed themselves so thoroughly through PANoptosis that the immune system is basically paralyzed.
So they have no defenses left.
Exactly. The patient is left completely defenseless against secondary bacterial infections that happen in the ICU.
Which brings up a massive clinical dilemma, honestly. I mean, if a patient comes into the emergency room on day 3 with a fever and dropping blood pressure, it could be 100 different things, right?
Oh, absolutely.
It could be sepsis, a weird flu, just a standard bacterial infection. How on earth do doctors differentiate a normal, albeit severe, infection from this specific runaway train of a cytokine storm?
Well, that diagnostic challenge is why the 2026 synthesis by Dr. Karki, Dr. Kanaganti, and their team is so vital. They highlighted specific, reliable biomarkers that clinicians can look for.
Okay, like what?
Well, one of the most fascinating diagnostic red flags is an extraordinarily high level of a protein called ferritin.
Ferritin. Okay, I know that as an iron storage protein. Like, if you are anemic, your doctor checks your ferritin levels.
Right.
Why is that suddenly the canary in the coal mine for a cytokine storm?
Under normal circumstances, yes, it just stores iron.
Yeah.
But macrophages, those heavy-duty immune cells we mentioned earlier, are absolutely packed with ferritin.
Oh, I see.
So during the intense hyperactivation of a cytokine storm, these macrophages begin pumping ferritin out into the bloodstream in massive, unnatural quantities.
How massive are we talking?
Well, normal serum ferritin levels might be somewhere between 20 and 300 nanograms per milliliter, depending on the person.
Okay.
In a severe cytokine storm, clinicians are seeing levels exceeding 5,000 nanograms per milliliter, sometimes even 10,000.
Wow. That is astronomical.
Right. When a doctor sees a ferritin level spike that aggressively, alongside dropping blood platelets and a fever, it acts as a massive, screaming red flag.
It tells them exactly what they're dealing with.
Exactly. It changes the entire paradigm of care. It tells the doctor, the virus isn't the primary threat anymore. The patient's own immune system is.
So what does this all mean for us? For the person in that hospital bed? If scientists have mapped the exact dominoes that are falling for, you know, the TNF, the interferon gamma, the Z-nucleic acids, the panoptosome, can we just, like, reach into the biological machinery and pull a domino out?
That is the ultimate payoff of understanding the mechanism. You see, for decades, the only real weapon doctors had against hyperinflammation was blasting the patient with high-dose corticosteroids.
Steroids.
Right, but steroids are a blunt instrument. They suppress the entire immune system indiscriminately.
Which the paper points out is incredibly risky. Because if you shut down the whole immune system with steroids, you leave the patient completely open to those secondary infections we just talked about. You solve the storm, but you lose the patient to pneumonia.
Exactly the risk.
Yeah.
But because this research has meticulously mapped the specific cytokine networks and signaling pathways like the interleukin-1, interleukin-6, and JK pathways, pharmacologists have been able to design targeted off switches.
Off switches. I like the sound of that.
Yeah, we use specialized drugs called biologics, which are basically engineered antibodies that intercept specific cytokines before they can bind to their receptors on the neighboring cells.
Okay, give me some concrete examples of these biologics from the research. How do they actually jam the signal?
Let's look at a drug called anakinra.
Anakinra.
Right. It is an antagonist specifically designed to block the interleukin-1 receptor. It sits in the lock, so the cytokine key can't fit.
Clever.
Then you have tocilizumab, which specifically binds to and blocks the interleukin-6 receptor.
Okay.
And beyond biologics, we have JK inhibitors, like baricitinib. JK pathways are the internal signaling wires inside the cell.
So what does baricitinib do?
It essentially snips the wire. Even if the cytokine hits the outside of the cell, the signal never reaches the nucleus. The cell remains deaf to the alarm.
Wow. So by strategically using a drug like tocilizumab, you are essentially putting earplugs in the neighboring cell.
Exactly.
It doesn't matter how much shrapnel or how many cytokines the bursting cell next door releases, the neighbor cannot detect the damage, so it never builds that PANoptosis guillotine. The loop breaks.
It breaks the cycle with surgical precision, allowing the rest of the immune system to remain online and completely functional.
And there is one specific application of these targeted off switches that is completely revolutionizing modern medicine. We have to talk about CAR T-cell therapy.
Oh, absolutely.
We hear about these miracle cancer treatments where doctors take a patient's own T-cells, engineer them in a lab with a kind of genetic GPS to hunt down a specific leukemia or lymphoma, and then put them back into the body.
Right. It's incredible science.
But the paper details a massive dark irony to this miracle cure.
Yeah, the irony is that the cure works almost too well. This is a condition specifically known as cytokine release syndrome, which is essentially a clinician-induced cytokine storm.
Okay, how does that happen?
When those engineered CAR T cells are reintroduced into the patient's bloodstream, they hunt down the tumor with terrifying efficiency. They latch onto the cancer cells and shred them rapidly.
But when you shred a massive tumor all at once, you're violently rupturing millions of cells simultaneously.
Which means you are flooding the bloodstream with an unprecedented volume of DAMPs, Z-nucleic acids, and cellular debris.
Right.
The engineered T cells also secrete massive amounts of interferon gamma to recruit help. And this massive debris field accidentally trips the ZBP1 sensors in the patient's healthy immune cells.
Oh, man.
Yeah. The panoptosis forms, the caspias activate, and the patient goes into a lethal cytokine storm caused entirely by the destruction of their own cancer.
The cure literally triggers the self-destruct sequence.
In the early days of CAR T-cell therapy, this was a devastating hurdle. You were curing the cancer, but the resulting cytokine release syndrome was pushing patients into lethal ARDS and multi-organ failure.
But this is where the 2026 understanding of the network changes everything, right? Because the doctors know the storm is coming. They engineered it.
Exactly.
So they can have these biological switches like tocilizumab locked and loaded.
It's a marvel of medical timing, really.
Yeah.
Oncologists administer the CAR T cells and let them begin their violent work on the tumor.
Right.
But they monitor the patient's biomarkers, like those ferritin and IL-6 levels, hour by hour. And the moment they see the threshold breached, the moment the positive feedback loop tries to establish itself.
They administer tocilizumab.
Yes, they block the IL-6 receptors, breaking the PANoptosis chain reaction in the healthy tissue, while the engineered T cells just finished destroying the tumor.
That is amazing. They let the security system catch the burglar, and the split second it tries to flood the house, they pull the plug on the water valve.
It is an incredible triumph of translational medicine. It perfectly illustrates why mapping the foundational biology of a disease is so vital.
Yeah.
By understanding the underlying unlytic mechanism of PANoptosis, we didn't just solve a mystery about viral infections, we unlocked the ability to safely administer some of the most potent, aggressive cancer therapeutics ever invented.
Okay, let's pull all of this together.
Yeah.
If we summarize this incredible scientific journey, there are basically three massive takeaways.
Okay.
Number one, cytokine storms are not just lingering chemical alarms. They are deadly, self-amplifying positive feedback loops driven by a violent, explosive form of cell death called PANoptosis.
Perfect. And number two, this single cascading cellular mechanism unites a vast array of completely different biological triggers. Whether it is a severe viral pathogen, a spontaneous autoimmune flare, or an advanced engineered cancer therapy, the underlying biological pathway of destruction is exactly the same.
And number three, by meticulously mapping these specific cytokine networks, the internal cellular sensors like ZBP1 and the executioner enzymes, science has given us the ability to intervene.
Right.
We can deploy highly targeted drugs like IL-6 blockers and JK inhibitors to pull the exact biological plug, stopping the storm without destroying the patient's innate ability to fight off disease.
It really requires us to completely reframe how we view critical illness. What we learn from Dr. Korkargan's Mercanoganti's synthesis is that lethality isn't always dictated by the strength, virulence, or toxicity of the invader.
That's a profound shift.
It is. More often than not, it is dictated by our own internal miscommunication. The line between a perfectly calibrated, life-saving defense mechanism and a catastrophic self-destructive attack is incredibly thin. It's really just an issue of balance and proportion.
It really is a razor's edge. And it leaves me with this thought for you to ponder. If we are finally mapping the exact circuitry of our own internal defenses and learning how to master the art of perfectly tuning our immune system's volume, turning down the lethal, fiery storm without completely muting our vital protections, what other currently incurable conditions might simply become a matter of flipping the right biological switches?
It's a great question.
If we can stop the AI security system from destroying the house, what else is the human body truly capable of surviving? Thank you so much for joining us in the Deep Dive Lab today. We hope you enjoyed this exploration into the human immune system's most dangerous mystery. Don't forget to follow and subscribe to uncover more fascinating science stories with us. Until next time, stay curious.